Okay, everybody, we are back with part 3. Okay, so lecture 7, bioenergetics. We're a little less than halfway through, so we're going to try and keep crushing through this. So, bioenergetics is the study of the energy changes during biochemical reactions. Specifically, we're going to be talking about reactions that cause energy to be absorbed versus pushing energy out. So this is going to explain why some of the reactions occur and others do not. Hello. Okay. We have the different biological systems are going to use chemical energy to power different living processes. We're also going to get a little bit into kind of exothermic versus, or endergonic versus exergonic. It's kind of referring to heat production. Remember, if we think about energy itself, energy cannot be created nor destroyed, only it can be transformed into another type of energy. So we have chemical bonds, and that's chemical potential energy. We make gradients, which are potential energy. There's kinetic energy, which is movement energy. And there's also heat energy. So all of these are going to be different types of energy, and we're going to see primarily we're going to go between chemical and heat. Okay. Well, chemical and heat, chemical to heat energy, and heat energy back to chemical. Okay, so we're going to talk about ATP, adenosine triphosphate. ATP is what allows for the coupling of thermodynamically unfavorable reactions to favorable ones. So hydrolyzing, so breaking apart ATP, releases energy, and that energy is then used to bind phosphorus to glucose. This is one of the examples. Then we have that first step in glycolysis. The phosphate binds to the glucose, and that's thanks to hydrolysis, and that is going to be a reaction that requires energy to take place. So the ATP comes, breaks the ATP, and by breaking the ATP, energy is released into that area, and then it's going to use that energy to then form a new bond. So the net sum is still negative, which is required for reactions to occur. So we can see our different interactions here. So like this, the 3.3 kilojoules per mole, or kilocalories per mole. This is telling us how much energy is required to form this bond. And this is how much, the negative is showing how much energy is released. Negatives is referring to energy being released and put out. Also, we can put it in the terms of heat, saying that a negative delta G is going to be exergonic and a positive is going to be endergonic. And we should always see the sum. The sum should always be negative for these different multiple step reactions. And it's negative because you have the energy coming from the breakdown of the ATP. So we absorb less energy than we release out in the total steps. There are also other high energy molecules that are used in this exact same way. The large molecules are broken down in this energy, and this will release energy. This released energy will be used to create new bonds between other molecules and atoms. The role of oxygen in energy production and utilization. Hopefully we've seen at this point why we need oxygen. We need oxygen in the Krebs cycle, and we need oxygen to be present in order for glycolysis to go from pyruvate to acetyl-CoA, otherwise it goes to lactic acid. And we also need oxygen as the final electron acceptor in the electron transport chain. Transformation of energy within biomolecules during biosynthesis. Biosynthesis means creating stuff. We also can call that anabolic reactions. An endergonic reaction means that it's absorbing energy, so it would get colder around the outside because it's absorbing the energy. And then also oxidative degradation, which is breaking apart, or catabolic reactions. That is an exergonic reaction that releases energy. And we also have transformation, which is a net exergonic reaction, releasing energy. And this will be going to be a pairing of endergonic and exergonic reactions to produce a product, which is what we actually see with here with glucose. We get the breakdown of ATP, exergonic, pairing it with an endergonic step in order to get the reaction to take place. The following concepts: Gibbs free energy. Talk about the energy that is available for doing work in a system. When we talk about a system is very concisely just we kind of in blocked off area that we're going to be looking at. A system could be, let's say, like the area around two specific molecules or one molecule itself, or an entire cell, or an entire organism, or the entire planet, or the entire universe. It just depends. It's kind of like you kind of set the scale of the system and determine like where, like whatever is present in that system. That's going to be your system size, and you determine like the energy stuff that's in that system based on what's there. There's standard free energy. This is the amount of energy released in the conversion of reactants to products under standard conditions. So concentration of 1 mole per liter at a pH of 7, and that delta G, the change in Gibbs free energy is going to be the delta G 0, a little space in on the apostrophe. There it is. We also have two concepts of enthalpy and entropy. Enthalpy is talking about heat content of the system. So if we increase the temperature in the system, we're going to increase the enthalpy, which increases the chances of reactions occurring. It's kind of like we're putting more energy into the system. As we heat it up, we put more energy into the system, it's more likely that reactions will occur. Entropy, which is called the chaos of the system. So it's the extent of disorder or randomness of a system. So even though we like to kind of pretend and talk that all of these different reactions and the chemical processes are very orderly and organized, there is a very big aspect of kind of randomness to it, where it's almost like everything is kind of floating around in this soup, and you're just kind of hoping that certain things hit each other the right way, they run into each other the right way, and when they run into each other, that's when these reactions will occur. So that's what entropy is talking about. If you have more stuff, more molecules, more particles, more stuff floating all around together and moving, so if there's more stuff moving around or more individual molecules that are moving around at once, there's more likely that more likely that something's going to run into each other, and more likely that a reaction is going to occur. That's what entropy is. It's the chaos of everything there. So more stuff packed into an area, the more chaos there is. And thinking about increasing entropy, entropy is typically increased by increasing the total number of moles. This can be done in a couple different ways. You can add more of the specific compound ever into the solution, or you can have that solution break apart. So if we take something like, if we're thinking like sodium chloride, right? NaCl. If you put NaCl, let's say we have 1 mole of NaCl. When you put that in water, it's going to dissolve, and you're going to get 1 mole of Na plus 1 mole of Cl. They're going to separate from each other. Now in total, you have 2 moles total, which means before you had 1 total, and now you have 2. Therefore, you have increased entropy or increased the likelihood of a reaction occurring. Hopefully that makes a little bit more sense about what entropy is. The different laws of thermodynamics. The first law is that the total energy of a system remains constant, that energy may be transferred or transformed, but never destroyed. Second law, that total entropy must increase if a process occurs spontaneously. So a spontaneous process, spontaneous reaction, one that just happens, has increased entropy. That means those spontaneous reactions are typically going to be degradation reactions, stuff breaking apart. Then there's these equations that relate them that are delta G, our Gibbs free energy, equals delta H minus the temperature multiplied by delta S. Okay, so the significance of free energy changes during biochemical reactions. What does delta G tell us? So it tells us if the reaction releases or absorbs energy. Sorry, let me put her in the other room real quick. Come here, baby. I know. Yeah. Girl. Okay, and we're back. So the significance of the free energy is going to tell us if the reaction is going to release or if it's going to require energy or absorb energy. And this is giving us the comparing that exergonic, releasing, or endergonic, which is absorbing or requiring energy. It also will tell us the amount of energy that is involved in the reaction and the ratio of the concentration of the products and the substrates at equilibrium. Here we have another equation. The relationship of free energy to equilibrium constant. Probably won't have much questions. The only kind of questions you'll have about this is like, which of the following is the equation for whatever. But what I find is that if you can understand the different components of the equation and then how they fit into the equation, it can really help you understand the overall concepts. So with the equations, I like to do this. I like to write out what all of the different things mean. The delta G, that was going to be our Gibbs free energy at standard values. Just kind of giving you the equation. And so you can pull stuff out of these equations. Like, for example, if you increase the concentration of the substrates, what does that do to the overall of everything? And you could plug this in, play with it a little bit, plug in some different numbers, and kind of see what pops back out, and you can kind of tell what would happen, what is actually happening with what would happen with specific reactions if certain things happened. Okay. Exergonic reactions. This is the relationship with free energy changes. So there's a negative free energy. Means the exergonic reaction heat is produced. So if delta G is very negative, the reaction goes to completion, which means that all the substrates are going to go into products. With really, really low or really, really negative number for delta G, it typically means that reaction is going to happen all the way. It's going to be irreversible, so it's not going to come back the other way. Because if it gives off a lot of energy, then it would require a ton of energy to go back into it to just happen by itself. And also, more than likely, that these really, really negative ones are going to be very spontaneous, meaning that they're less likely to need stuff like enzymes, less likely to need like these very perfect conditions or help in order to react. It's just going to happen, and it gives off that heat. And this is our catabolism, catabolic reactions. Endergonic reactions are essentially the opposite, so that they're a positive free energy. It's absorbing energy. The delta G is very positive. That's saying that the system is stable and it will not occur spontaneously. These reactions are not going to just happen. They're going to need some sort of assistance. Like, glucose does not just become phosphorylated on its own. There has to be an ATP, and there has to be an enzyme to come and to help it. So that in this case, the hexokinase or the glucokinase needs to come to help activate that process, lower the activation energy, in order for that reaction to occur. And those are our anabolic reactions, also called synthesis reactions. In the equilibrium conditions, what do we see? So the rate of forward reaction should be equal to the rate of backwards reaction. The rate of the backwards reaction is equal to the rate of the forward reaction. It's just saying the same thing. And free energy changes, there should be no net change of free energy. Once we're at equilibrium, everything should be constant. Everything should be equal to one another. The significance of the delta G values and its relationship with KEQ. KEQ is our equilibrium constant. So it's the ratio of the mathematical product of the concentrations of the products of a reaction to the mathematical product of the concentrations of the reactants of the product. So it's comparing those reactants and the products. So for example, if our and so this is showing that, so once we're at 1, KEQ of 1, that means that we are at equilibrium, and that's typically what our body will eventually, most of the time, wants to get to, is at an equilibrium point. But so if our delta G is greater than 0, meaning it's very, very positive, our KEQ is going to be less than 1. So the reaction is driven backwards. Endergonic reactions. If it's more than, it's going to be exergonic. It's going to go forwards. So forwards meaning from reactants to products. This is talking about the coupling of exergonic and endergonic reactions in biochemical reactions. Really, if you think about this should be pretty straightforward, where if you have a reaction that's not favorable, that you need to input energy into in order for that reaction to occur, that energy has to come from somewhere. So if you can pair it with an exergonic reaction, that's going to give you the energy that you need to then create that endergonic reaction for everything to come down and to happen. So pairing them, just especially in the body, just sort of makes sense to us in order to get to the point where, or in order to be able to have those reactions happen. Activated molecules that gain energy for synthesis or breakdown of molecules. I didn't have any written down here. I'm sure there might be a couple of examples in the book. I didn't have any examples specifically that I had put down. But there are a couple that lose energy for synthesis, like adenylate cyclase and the nucleoside diphosphate kinase. These next pictures are just kind of showing those specific reactions. This is again just going over how the exergonic product releases energy, making it available for the endergonic reactions, very similar to what was just above. And that gets us through the biochemical, bioenergetics, that's what it says, the bioenergetics. On to lecture 8, Introduction to Carbohydrates. Hopefully at this point you have mastered carbohydrates, because we've talked about them a lot. So these are the most abundant class of biomolecules and essential for all living organisms. Carbs are required for life, okay? Just my quick little jab at the keto diet. If you've tried it and it's worked for you, great. But just to be clear, doing a keto diet long term, according to the science, not myself, I'm not giving any recommendations personally, but according to the research, the keto diet is not a long-term sustainable diet. And if at any point in the future, if you're in an office and it is within your scope of practice to give nutritional advice, hopefully at that point you will have learned enough about this to look at other options that could be very beneficial to patients. I'll leave it at that. Okay, so the importance of our carbohydrates is both structural as well as metabolic. So glucose obviously is our metabolic side. All of our good stuff that glucose does, that we talked about, that we burn into being stored for energy, using the a pentose phosphate pathway to be converted for nucleic acid synthesis, lubrication of joints, signaling molecules, all sorts of stuff that glucose is used for. The classifications of carbohydrates, we have the aldehydes and the ketones, the monosaccharides, there we go. The monosaccharides, the aldoses, and the ketoses. All the structures of the monosaccharides. Don't spend a ton of time on this. Maybe brush up on the aldoses and the ketoses. Just like the ribose versus ribulose, glucose and versus fructose. Could throw a question in there about that, but again, the gross memorization isn't always what will be tested. But yeah. Okay. We also have disaccharides, oligosaccharides, and polysaccharides, 7 or more monosaccharides joined together. Stereoisomers, this is just some definitions where they have the same structural formula but different in their spatial arrangement. Isomers, the presence of asymmetric C or carbon atoms allows the formation of isomers. D-isomer, again, just knowing that the D-isomers are what are found primarily in the body. Pretty much everything is the D-isomer. I think there's one that's an L, but they're all pretty much D-isomers. Okay, we got a whole bunch of pictures coming through here of all our different ketoses, all this sort of stuff. Again, this just wasn't super. You can go through this, but to be honest, just review it, and kind of, specifically the ones that are boxed in. But to be honest, this was kind of that one chapter. These won't come up a ton. Just as like raw memorization, they won't really come up. They shouldn't come up a lot for the exam. Polysaccharides, again, just a whole bunch more names. I would remember chitin, remember cellulose, amylopectin, and amylose. I remember those guys. The fact that starches are in plants, it's a plant storage form of glucose. We have glycogen, they have starch. Yeah, that's about it. Glycosaminoglycans, these are components of the extracellular matrix, so kind of the structure of the cells. And they're complex polysaccharides containing amino groups. The glycoconjugates, these are kind of the fused together forms of glucose, where you have protein with glucose, glycolipids, and glycoproteins. Oligosaccharides, linkage in glycoproteins. I'll be honest, I don't really remember talking a lot about this, but there are the different types of linkage. To glycolysis. Doing pretty good on time. Okay. Again, hopefully you have beaten glycolysis through your skull 8 million times at this point. Get ready for number 8,000,001. So the role of glucose in metabolism. So that's going to provide the fuel for physiological brain function through the generation of ATP. Glucose can go several different directions. You can go into the storage form. It can go into the oxidation via glycolysis. It could also go into the pentose phosphate pathway. Or it can go into the making of the different structural type stuff for the body. So glucose is very, very versatile. We need it for lots and lots of reasons. We're talking about the amphibolic pathways of glucose metabolism. It's the biochemical pathway that involves both catabolism and anabolism. So what is glycolysis? The glycolysis is the breakdown of glucose by different enzymes. It's going to release energy and pyruvate This is going to be the whole purpose of glycolysis, is to create a little bit of energy. Again, it creates 2 net ATP per glucose. It creates a total of 4, but we use 2 to get it there, so it's a net of 2. We also make 2 NADH and 2 pyruvate. So the whole point is to make pyruvate. Making pyruvate allows us to turn it to acetyl-CoA to be put into the Krebs cycle to then make more NADH and FADH2 in order to go into the electron transport chain to make a whole bunch of ATP. That's the whole point of glycolysis, is for energy. Remember that if we don't have oxygen present, the pyruvate, or the NADHs that were created during glycolysis, break down and react with the pyruvate, and they form lactic acid. There's 10 total enzymes that we'll go over. So the sequence, we have our first chunk is our priming phase. Glucose hexokinase adds a phosphate to glucose, glucose 6-phosphate. Our phosphohexose isomerase, it isomerizes glucose to get us fructose 6-phosphate. Then we have PFK1. Sorry, I got a text from my wife. Okay. The PFK is going to be their second priming reaction. These priming reactions are going to be very, very important. These are 2 of our rate-limiting steps. We need to remember those. Those are going to be the ones that most likely we're going to get asked questions about. That adds another phosphate, so we get fructose 1,6-bis biphosphate. And then the aldolase is going to cleave our 6-carbon glucose into 2 3-carbon sugar phosphates. We get glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Dihydroxyacetone phosphate goes off to the side, does its own little thing, and then gets converted to glyceraldehyde 3-phosphate as well. So now we have 2 of them. We continue, we pump out some ADH with the dehydrogenase. Remember, the dehydrogenases give us what are going to give us our NADHs. Then we have phosphoglycerate kinase. The kinases are dealing with phosphates, phosphorylation. So it's going to give us our first 2 ATP, bring us to neutral. Then we have a mutase, an enolase, and then finally pyruvate kinase, which gives us that last one to finally create pyruvate. Those are 10 different steps. We'll go review back through glycolysis a couple of times. If you can draw it back out. You don't have to memorize like the full entire name. I would abbreviate a lot of these, like hexokinase. If you can remember, like hexo-K or phosphohexose isomerase, PHI, phosphofructose PFK, aldolase, if you just remember A, or Aldo, something like that. It'll help just remember all of that. Okay, and there's that whole thing all put together. The dihydroxyacetone phosphate, the enzyme that acts on that is the triose phosphate isomerase, which is going to reorganize it and turn it into a glyceraldehyde 3-phosphate. The enzymes, the enzymes are actions that catalyze the intermediates used and the products produced in the pathway. Again, that's this whole thing. We have the hexokinase or the glucokinase. Remember, glucokinase is what is found primarily. We find that in the liver. Hexokinase is going to be in the muscles. Yeah, and I have all this. Like, if you want to do this as practice, you can. Personally, I'd recommend though just sticking to drawing this out. Be able to draw it out a couple of times with everything abbreviated, and you'll be fine. The metabolic fate of lactate as gluconeogenic precursor. So lactate, or lactic acid, is oxidized to pyruvate, and NAD plus is reduced to NADH and H plus. These proceed through aerobic metabolism or oxidation. Lactate circulates to cardiac cells. During heavy exercise, lactate becomes the preferred fuel of the heart. So, when we don't have enough oxygen, our body converts everything to, well, not everything, but it'll start to convert some of the pyruvate that we're making into lactate. That lactate, this is what this is saying, is that it is going to get used up because the lactate can be oxidized back to pyruvate and the NADH. So it's not stuck there permanently. It can be brought back out and then used for energy. Now, lactoacidosis is an issue when the lactic acid builds up in the bloodstream, so we have too much lactic acid. And if you think about it, if it's an acid, you put an acid into a solution that is primarily water, which blood is primarily water, what happens to the acid? Acids are going to make things more acidic. They're going to give off hydrogen atoms. Giving off a hydrogen atom is going to do what to the pH? It's going to lower the pH, which is a no bueno because the pH should be very specific in the blood, 7.35 to 7.45. I've said that enough. Please remember that number. You will need it for physiology. With glycolysis, we have the three main enzymes, and these are the highly exergonic and are reversible: the hexokinase, the phosphofructokinase. Actually, hold up. Sorry, and are irreversible, highly irreversible. I think I misspoke there. Hexokinase, phosphofructokinase, and pyruvate kinase. Know those three, especially for boards. Really remember these three because you need to know those guys. You'll get at least one question, most likely about phosphofructokinase. Okay, lecture 10. TCA cycle or citric acid cycle or the Krebs cycle. These are all AKAs for the same exact thing. So again, going through it. So pyruvate is oxidatively decarboxylated to acetyl-CoA by pyruvate dehydrogenase complex, PDC. So the pyruvate dehydrogenase complex has three different enzymatic activities. You have the pyruvate dehydrogenase, the dihydrolipoyl transacetylase, and the dihydrolipoyl dehydrogenase. The pyruvate dehydrogenase E1 catalyzes the decarboxylation of pyruvate and forms an acetyl group. COO, CO2. Again, pulling that off, it's going to be pulling off a CO2, and that's where we get one of the stages where we get CO2 from during the creation of energy. The coenzyme is the TPP that helps with the decarboxylation and CO2 formation. The next part, the E2, catalyzes transfer of the acetyl group to CoA-SH, and we have the coenzyme A as the coenzyme. And then the next part is going to catalyze regeneration of the disulfide form of lipoate. The electron passed to FAD and then to NAD+. So we get passed through a couple different things that we use in the third step. These are the coenzymes used in this one, used in the PDC. Big ones to remember are thymine pyrophosphate, of course, NAD and FAD, coenzyme A. Lipoate, occasionally you'll see a question on, but not as common as like TPP. TPP typically is one of the favorites to be asked about. Did you know the regulatory role of PDC in the conversion of pyruvate to acetyl-CoA and know the importance of PDC in the pathway? First and foremost, without PDC, we don't get Krebs cycle. If there's no way to convert pyruvate into oxaloacetate, then you're not going to get the Krebs cycle to happen because oxaloacetate, where's my brain? Pyruvate goes to acetyl-CoA. Yeah. Sorry, brain had a little bit of brain fart there. We're good. So it goes to the acetyl-CoA. If the pyruvate does not go to acetyl-CoA, then it does not go into the Krebs cycle. Okay. Also the PDC needs to be regulated, and this is regulated by we're going to call it essentially negative feedback, where once we have a lot of the products that are formed through the chain of PDC, all of these guys, it's going to shut it off. And it's done through this by phosphorylation of the PDC. It shuts it off. Activation, again, if we have a lot of these here, so if we don't have ATP, if we don't have acetyl-CoA, if we don't have NADH, etc., then we're going to want to activate PDC in order to get those. So the sequential reaction of the citric acid cycle with special reference to, I'm pretty sure it's like the reaction types and what's created. Let me kind of skip all down through here until we get to, do I have I guess I don't have the actual picture here of the Krebs cycle. Let me pull that up real quick. And again, this right here, these are my part 1 board notes. Here we go. These are my part one board notes, along with, I made a Quizlet for biochem as well that you can find. You can find these notes again available in the link in the description. Okay, so we're following the pathway. Acetyl-CoA comes in and goes to get actinamide citrate synthase into citrate. We get cis-aconitate, isocitrate, alpha-ketoglutarate, to succinyl-CoA, to succinate, to fumarate, to malate, and to oxaloacetate. Remember the mnemonic for this: couples cuddle insanely often after special sex for marriage obligations. And also remember this little one where I am the most important. So at I, A, and M. So at the isocitrate, the alpha-ketoglutarate, and the malate, that is where our NADH get formed. And you can also think, if you want the FADH2, gets right before the fumarate, before the other F. F goes with F. Okay. You can go through and kind of look at all of the different steps and reactions. I'll leave this on the screen for a second. Again, this is very similar with glycolysis. Draw this a couple of times. Mainly just focus, don't worry so much about like what's coming off, like these green and these red things coming off here. Focus on where the NADH comes out, where the FADH, where this GTP is. And familiarize yourself again with the specific enzymes and kind of what they do. Kind of, are they like decarboxylation? What does decarboxylation do? What does phosphorylation mean? Dehydrogenation, except all of that sort of stuff. Focus on those things. Production of reducing equivalents. That's exactly what I said. I am the most important. Production of the high energy phosphates, we get 2.5 ATP per NADH and 1.5 ATP per FADH2. Know that the dehydrogenase enzymes are responsible for NADH and FADH2 formation. If it's a dehydrogenase enzyme, the dehydrogenases are what's going to form the NADH and the FADH2. The three enzymes that regulate citric acid cycle and the regulation mechanisms: citrate synthase, isocitrate, alpha-ketoglutarate dehydrogenase. These are big ones that we want to remember. So these three regulatory enzymes catalyze the highly exergonic reaction and lead to the irreversible reactions that energetically favor formation of a product in a forward reaction. So just like in glycolysis, these are our rate limiting steps and what determine kind of how quickly we're going through everything and whether the reaction is continuing or not. And this is all, again, a bunch of repeat. You can pause and look through this, or if you want, the link is in the description. You can get this whole document already all put together, and so that you can go through and highlight and add different stuff of your own, rather than having to type the whole thing up yourself. Know which enzyme catalyzed reaction produces high energy phosphates, such as GTP. We wouldn't talk about that. How many ATP are produced from one NADH and one FADH? We talked about that. The role and significance of the citric acid cycle in metabolism. We make the NADH in preparation for electron transport chain. The citric acid cycle intermediates. This is this whole chart. Again, you want to mostly focus on oxaloacetate, as this is the most common one. Kind of all of the arrows going in and away from oxaloacetate, I would mostly focus on those. But understand, like, there's the malic enzyme that can turn pyruvate into malate. So there's different ways for these to turn into the intermediates or be used for other purposes, for other biosynthesis purposes. Citrate goes to fatty acids and sterols. We'll learn more about that in the first couple units of Biochem 2. Alpha ketoglutarate, we can get our purines. So, and I would focus mostly on those. Again, oxaloacetate. I would know pyruvate to oxaloacetate to the phosphoenol pyruvate, 3-PEP carboxylase. Pyruvate to malates, citrate to fatty acids, and alpha ketoglutarate to purines. Those are the ones that I would remember. Know how they are regulated. Really, I have written here like what causes everything to activate versus inhibit for each individual step. But in general, like if there's a whole bunch of the next intermediate, it's going to be inhibited, and if there's a bunch of the intermediate before it, it's going to be activated, as well as like if it's one of the steps that produces NADH or FADH2 or GTP, if there is a lot of that already, it's going to be inhibited. If there is the precursor, so if there is a lot of NAD plus or just FAD or GDP, for example, then those reactions are going to be kind of more likely to occur. They're going to be activated rather than spending all this time memorizing all this stuff. Yeah, don't worry about that as much. Know the reasoning behind it, not necessarily just for memorization. Okay. This formula, I have here written here, know this formula. I would recommend knowing it. If you don't try and sit down and memorize it, again, understand the concepts, what this is saying. Acetyl-CoA plus the 3-NADH, 3-NAD plus FAD, GDP, phosphorus, 2H2O gives you all of this. This is the entire formula for the Krebs cycle, kind of what goes in and what comes out of the Krebs cycle. Okay, and we'll go ahead and stop this part here, and we'll jump into the next part. We're doing pretty good. 53 of 85 pages, so we're flying.